How's it going, everyone? It's Chow here, and today we are going to talk a little bit about respiration, so let's get started. Okay, so respiration is the active exchange of gases in specialized structures, and we're talking about this exchange of oxygen and carbon dioxide within the body. We can explain this with a little bit of physics, more specifically with Fick's law of diffusion. So we have the Q value, which is basically the rate of diffusion, and we also have partial pressure. So partial pressure is the hypothetical pressure of a gas in a mixture of gases, such as air, if that particular specific gas were to occupy the same space by itself. So this is the partial pressure over here. And if we increase the partial pressure, right, P1 minus P2, and we increase that value, so maybe we make P1 bigger, and we make the partial pressure larger, we're actually going to increase the rate of diffusion. In addition, if we actually increase the surface area, which is the A value over here, we're also going to increase the rate of diffusion. So that should be something that's supposed to come pretty naturally to you at this point. Increasing surface area allows for greater exchange if we're talking about gases or diffusion or something like that. In addition, we actually have the ability to increase potentially membrane permeability. So this is related to the D value, which is the permeability coefficient. And if you increase the membrane permeability, you actually increase the rate of diffusion. That should be pretty straightforward. And finally, we have this over here, which is basically your membrane thickness. So it's kind of like the path length. And if you actually increase the L value over here, so you're increasing the thickness of the membrane, you're ultimately going to decrease the rate of diffusion. Whereas if you decrease the membrane thickness, you decrease the L in the denominator, that's going to make this number bigger and therefore increase your rate of diffusion. If you make this L larger, this number is going to be smaller, so you're going to decrease the rate of diffusion. So that's basically what the L stands for, and this is a general rundown of Fick's law of diffusion. A little bit more on that equation, though. So this equation, this Fick's law equation, really just reflects on the adaptations that have evolved within different lineages of organisms to increase respiratory gas exchange. So what really is trying to do is minimize the diffusion path length by making thinner tissues in gills or lungs, or sometimes tracheae, or internal gills. So we're looking at, for instance, external gills on the salamander. You want to minimize the path length difference and make these relatively thin to allow for easier passing of gases. Same thing for within certain tissues within the lungs. Also, what you can do is you can maximize the partial pressure, again, which is over here. You maximize the partial pressure gradient by decreasing the partial pressure in the tissues. So by shifting the difference in the partial and the pressures between the tissues and surroundings and the outside and in the cavity, for instance, you can actually increase, right? You can increase the ultimate partial pressure difference, and that can ultimately increase this number over here. Remember, partial pressure is in the numerator. And finally, you can maximize the amount of diffusion by circulating the blood and basically actively moving that air and water around, which should make pretty straightforward sense, I'd say. And finally, last but not least, I guess, also is you can increase the amount of surface area available. So by increasing, again, the surface area, you can also allow for that uptake of the rate of diffusion. So you can increase the A value and then also ultimately increase the Q value, which is your rate of diffusion based on Fick's law of diffusion. Okay, so what we're talking about, that movement of air from the outside world into our lungs, we are really talking about ventilation, not respiration. Again, respiration really is only specifically for that exchange of gases between the membranes. So we're looking at tidal ventilation. So air is going to be flowing in and out of the lungs in mammalian species via the same pathway. So this results in a residual volume that is basically never able to be completely exhaled. There's always some leftover air in your body. What that means is that every time you inhale, that old air is going to be mixing with fresh air and ultimately reducing the partial pressure of that oxygen. So to kind of ameliorate and attenuate these problems, this effect of partial pressure, so you want to reduce the effects of this, you can actually have a larger surface area in the lungs to kind of mitigate the issue. And then you can also decrease the path length difference. So you can also decrease the membrane thickness, again, decreasing L. So these are ways you can help reduce this issue over here. Okay, so you can measure this lung ventilation using a respirometer. And this is the chart that you will often see in a respirometer, and this is also, I think, in your textbook. So there are a couple of things that you can look at. The first one is your tidal volume, or TV, over here. And this is your normal volume when you're at rest, which is pretty straightforward. And then you also have inspiratory reserve volume, IRV, and this is the additional volume of your deepest inhale. So what's your deepest amount that you can inhale into your lungs? You also have expiratory reserve volume, or ERV, which is shown over here, and this is the additional volume you can forcefully get out of your lungs. You also have the residual volume, which is shown over here, and this is the amount of air in your lungs after the maximum exhale. So this is the amount of air that stays in your lungs even after you forcefully exhale air. So there's always a little bit of leftover air in your lungs if you're a healthy individual. So finally, you can look at some of these combined elements, which include vital capacity, which is basically your tidal volume over here, plus your inspiratory reserve volume, as well as your expiratory reserve volume. So it's this over here. So this is vital capacity. And then you also have functional residual volume, which is just basically your ERV plus your RV down here. And finally, if you add everything up, that's going to be your total lung capacity. So this is a way you can help measure, right, your lung ventilation. Okay, so the path of air in the lungs, I'm not going to talk too much about this. The lungs are in the thoracic cavity. That should make a lot of sense. You have this thing down here. It's a sheet of muscle called the diaphragm, and basically by contracting or relaxing, it can help you ultimately breathe in and out. You also have a pleural membrane that covers each lung and lines the thoracic cavity. So in terms of the air coming in, it's going to go through your nasal or oral passage. It's going to enter into your pharynx and then eventually enter into your larynx. It's going to go into your trachea and branch out into the bronchus or the bronchi. The bronchi will then branch out into little bronchioles, which eventually reach your alveoli. And it's the alveoli where your actual act of respiration or actual exchange of gases occurs. So there are kind of two things that are very important in this path of air in your lungs. The first one is negative pressure. So what negative pressure is the slight pressure that occurs in the pleural cavity when the volume of the thoracic cavity increases. So that occurs when you're inhaling. This negative pressure that's present basically keeps the alveoli inflated even between breaths. So you're not going to deflate the alveoli, and that negative pressure can help maintain that inflated alveoli. In addition, there are also surfactants in the lungs, and a surfactant is basically something that reduces the surface tension of a liquid. So it's going to give that liquid more flexibility. And in your lungs, there is kind of a fluid inside the alveoli with reduced surface tension, which allows the lungs to stay elastic and reduces the amount of energy needed to make them inflate. So you could think about this maybe in a way where your lungs are made of maybe like wax paper rather than tissue paper. So when you have certain things flowing through it, when you have certain moisture that's around it, the surfactant basically keeps it relatively stable and open rather than cause it to collapse. And these surfactants, I believe, are generally made of phospholipids, sometimes ions, as well as certain membranes that are secreted in the alveoli. So surfactants can help out with your breathing in addition to that concept of negative pressure. And then finally, we have the alveoli, which is basically the site of gas exchange in the lungs. These alveoli are coated with very thin capillaries to allow for that gas exchange. So remember, gas exchange really only occurs within our capillaries. So it also has a thin membrane, so remember you're decreasing the thickness of that membrane, you're decreasing that L value, which is shown over here, and by decreasing that L value, you're going to have the small path length difference and therefore increase your rate of diffusion. In addition, the alveoli are very small, so you're trying to maximize the surface area to increase the amount of diffusion. So you can fit more surface area into a smaller amount of space. Also, the oxygen will go into the alveoli, is then picked up by the red blood cells in the capillaries, and then is eventually pushed on to where it needs to go. So this is the site, the alveoli is the site where that exchange again actually occurs. So we talk about ventilation and breathing, but the actual respiration, where the site of gas exchange actually occurs in the alveoli. So you have that oxygen that goes into the alveoli, it's picked up by the red blood cells in the capillaries, and then that carbon dioxide is dropped off, and you can go ahead and exhale it afterwards. Now that the blood is charged with oxygen and is oxygen rich, you can pass that blood from the lungs into your left atrium, and then into your left ventricle, and then into your aorta, and that can therefore go into the systemic loop and be passed on to wherever your body needs oxygen. So this is just a short video on information in regards to respiration from a physiological level. I hope you found it useful, and best of luck studying, and I'll see you in the next video. And we're most likely going to discuss a little bit more about hemoglobin and blood as a whole.